Hello students, welcome to the class of Bioinformatics. Today we will discuss semi-global alignment, the Smith-Waterman algorithm, and we will do some problems. The basic alignment algorithm discussed so far performs a global alignment. That is, it compares two sequences in their entirety. The gap penalty is assessed regardless of whether gaps are located internally within a sequence or at the end of one or both sequences. This is not always the most desirable way to align two sequences. Suppose we wish to search for the short sequence ACGT within the longer sequence AACGTGTCT. Of the several possible alignments between the two sequences, the one we are most interested in is AAAACGTGTCT, gap 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 ACGT, gap 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 gap. This is the most interesting alignment because it demonstrates that the shorter sequence appears in its entirety within the longer sequence. When searching for the best alignment between a short sequence and an entire genome, we might wish to avoid penalizing for gaps that appear at one or both ends of the sequence. Here, the terminal gaps are usually the result of incomplete data acquisition and do not have biological significance. This approach is referred as semi-global alignment. Fortunately, we can conduct such an alignment by making a few small changes to our basic dynamic programming algorithm. So let us understand the difference in semi-global alignment compared to Needleman-Wunsch algorithm. In Needleman-Wunsch algorithm sequence alignment, a vertical move represents a gap in the sequence along the top axis, and a horizontal move represents a gap in the sequence along the left axis with penalty value for the gap. But semi-global alignment do not want to compute penalty for the terminal gaps. So leftmost column and top row of the matrix will be initialized with the value 0. Additionally, horizontal move in the bottom row and vertical move in the rightmost column are penalty free. The difference in initialization of semi-global alignment and Needleman-Wunsch algorithm is demonstrated here in this figure. Please note, the difference is not only in the initialization of the first row and first column, but in the last row and last column, the horizontal move in the bottom row and vertical move in the rightmost column are also penalty free. Here in the figure, the leftmost column and top row of this partial alignment score table have been initialized with the value 0. Additionally, horizontal moves in the bottom row and vertical move in the rightmost column are penalty free. This allows gaps at the start or end of either sequence to remain unpenalized. The arrows indicate all allowed paths from the lower right corner of the partial score table to the upper left corner, as in Needleman-Wunsch algorithm for alignment. So I hope you got the concept about semi-global alignment. Now let us understand an algorithm for local alignment, Smith-Waterman algorithm. Sometimes even semi-global alignments do not afford the flexibility needed in sequence search. For example, suppose you have a long sequence of DNA and you would like to find any subsequences that are similar to any part of the yeast genome. For this sort of comparison, a semi global alignment will not suffice since each alignment will be penalized for every non matching position. Even if there is an interesting sequence that matches part of the yeast genome, all of the non matching residues are likely to produce an absmite alignment score. The appropriate tool for this sort of search is a local alignment, which will find the best matching subsequences within the two search sequences. The Smith-Waterman algorithm can be used for local alignment. With minimal modifications, our dynamic programming method can be used to identify subsequence matches while ignoring mismatches and gaps before and after the matching regions. The resulting algorithm was first introduced by F. Smith and M. Waterman in 1981, which is a fundamental technique in bioinformatics. So now let us understand how the Smith-Waterman algorithm can be used. Filling the table is similar to global alignment algorithm, Needleman approach, but we place a 0 in any position in the table if all of the methods result in scores lower than 0. Once table is completed, we identify the maximum partial alignment score, then work backwards as global sequence alignment and construct alignment until we reach 0. Now, to understand this Smith-Waterman problem, to find the best local alignment, let us do a problem. Two sequences are given. Let us understand the Smith-Waterman algorithm by doing one problem. Consider two sequences. First sequence is A A C C T A T A G C T. We initialize the with zeros. Second sequence is G C G A T A T A. We are using Smith-Waterman algorithm for the local alignment of these two sequences. Gap penalty value is -1, match bonus score is +1, and mismatch score is -1. As designed in the Needleman-Wunsch algorithm, we form m plus 1 rows and n plus 1 columns. In semi-global alignment, we do not want to compute penalty for the terminal gaps. So the leftmost column and top row of the matrix will be initialized with value 0. Filling the table is similar to global alignment algorithm. For remembering the values I am writing here, gap is -1, match score is +1, no match score is -1. When we take the diagonal elements or left values or computation values from the top, if the result is negative, we write 0. We do not write negative values in the cells. We proceed like this. When match score occurs for the diagonal elements, we write 1 by adding the diagonal element value. Here, when we take the value from the left side 1, we have gap value -1. So 1 plus -1, value becomes 0. Thus we fill all the values in the cell. We place a 0 in any position in the table if all the methods result in scores lower than 0. All other computations are similar to Needleman-Wunsch algorithm method. Once the table is completed in this manner, we find the maximum partial alignment score in the entire table, which is 4 here. The maximal value in the partial alignment score in the table is 4 here. Starting with this position and working backward until we reach a value of 0, we obtain the alignment. We choose the alignment sequence based on the diagonal arrows, as we discussed in Needleman-Wunsch algorithm. Here the best local alignment sequences are TA, TA, and best local alignment score is 4.